So I want you to picture a sound. It's a sound that literally nobody ever wants to hear. But it happens, about 300,000 times a year in the U.S. alone.
Right, the alarm.
Yeah, the alarm. It's like 2 a.m. You're in a bustling emergency room in some major city. A patient gets rushed in on a gurney. And just, minutes ago, they were talking, breathing, living their life. Their heart was doing that incredible thing it does, pumping, sustaining life. But now
it's just chaos.
Complete chaos. You look up at the monitor, and it's not a flat line. I mean, that's kind of just a movie myth, right?
Yeah, totally a movie myth.
It's actually this violent, ragged scrawl on the screen. The doctors, they call it ventricular fibrillation.
Yeah.
The main pumping chambers of the heart, they aren't pumping anymore. They're quivering.
Like a bag of worms. That's the clinical description often used. A bag of worms.
Which is horrifying.
It is. And this is the critical moment, right? You have minutes, maybe just seconds, before the brain starts to die from a lack of oxygen because the blood has completely stopped moving.
Yeah.
So what is the standard of care? What is the tool we immediately reach for?
I mean, we all know this one. It's the absolute staple of every medical drama ever made. The paddles.
Exactly.
The defibrillator.
Clear. And then zaps.
And look, it works. It saves lives every single day. But have you ever actually stopped to think about what we are doing to the human body in that exact moment?
Well, we're shocking it, right? We're jump-starting the battery.
See, that's the thing. We are doing so much more than just jump-starting a battery. We are delivering 200 joules of energy directly into the chest cavity. And to put that in perspective for you, that is enough electricity to arc weld sheet metal.
Yikes. I knew it was a kick, obviously, but arc welding. Yeah. It is a massive brute force assault on the entire system. And in the world of complexity science, which is, exactly what we are diving into today, we have a very specific name for this approach. We call it the sledgehammer.
The sledgehammer?
We use the sledgehammer because for basically 50 years, we've been trying to open a locked door, and we just didn't have the key. We didn't understand how the lock works. We just kicked the door down. We jolt every single cell in the heart into submission simultaneously because we don't understand the specific shape of the chaos that's actually killing the patient.
Wait, the shape of the chaos? That's a weird phrase. I mean, chaos isn't supposed to have a shape. Chaos is messy. It's random. It's literally the opposite of having a shape.
And that right there is the massive misconception we are going to completely dismantle today. I want to take you away from that ER for a second. Let's travel back to 1992.
Okay.
A quiet laboratory at UCLA.
Yep.
No panic, no screaming doctors, just the low hum of a computer.
I'm there. What are we looking at?
The researchers there, Alan Garfinkel and his team, are looking at a tiny piece of heart tissue. It's rabbit heart, actually, about the size of a postage stamp. It's sitting in a little dish of warm saline. And just like that patient in the ER, this tissue is fibrillating. It is in total chaos.
So what, they get out the mini paddles, the mini sledgehammer?
No, and that is the absolute breakthrough here. There are no paddles. Instead, they have a computer watching the chaos. It's analyzing the electrical waves in real time. It's plotting the geometry of the disaster and just waits. It calculates. And then, at precisely the right millisecond, it delivers a pulse.
Like a big pulse.
A whisper. Millivolts. Barely enough electricity to tickle a nerve.
And what happens?
The chaos snaps back into perfect order. Immediately.
Whoa.
Not by overwhelming the system, but by understanding it, by just nudging it.
That feels like literal magic.
It's not magic. It's geometry. And that brings us to the real core mission of this deep dive. We're all taught that health is order and disease is chaos. We think the healthy heart is a perfectly ticking metronome, and the sick heart is a storm.
Right. Order equals good, chaos equals bad. That's like the most basic rule of biology.
But what if that's entirely wrong? What if the shape of chaos is actually the very thing that keeps you alive? What if losing your chaos is the real danger?
Man, I love an inversion. Let's really get into it. Welcome to the Deep Dive, everyone. Today is all about life on the boundary. We've got an incredible set of sources to pull from, and our mission today is to figure out how chaos theory actually applies to the flesh and blood inside you.
I am so ready for this. This is the application phase. Last time we talked, we built the mathematical tools, and today we actually use them to understand life itself.
Yeah, and before we start, hacking into the heart and the brain, we probably need to do a little housekeeping. We did a deep dive recently on the foundations of all this.
Right, the load-bearing concepts.
Exactly. So if you heard that one, consider this a quick refresher. If you didn't, don't worry. We are going to give you the clean cocktail napkin version right now. Because if you don't grasp these a priori concepts, the rest of this deep dive is just going to sound like complete sci-fi babble.
Fair enough. Where do you want to start?
Let's start with the billiard balls, the whole concept of deterministic unpredictability.
Perfect. So this is the idea that basically destroyed the old Newtonian view of the universe. In the old days, scientists thought, hey, if I know the position of every single particle in the universe, and I know the laws of physics, I can predict the future perfectly.
The clockwork universe.
The clockwork universe.
Yeah.
But then came the billiard balls, courtesy of mathematicians like Jacques Hadamard and later Edward Lorenz with weather. Imagine a pool table. If I hit a ball and there's no friction and I know exactly how hard I hit it, I can predict exactly where it goes. It's easy.
It's just geometry 101.
Right. But if I add a second ball and a third, and they start colliding, it turns out that if I make a tiny mistake in my measurement of that very first hit, an error as small as, say, 1/1000th of a degree after about 8 collisions, my prediction is completely wrong.
Because the error compounds.
It amplifies exponentially. This is the famous butterfly effect, sensitive dependence on initial conditions. You can know all the rules, meaning the system's deterministic, but you absolutely cannot predict the specific path it will take. The trajectory is lost to you.
Okay, so we can't predict the path. I mean, that sounds like terrible news for a doctor trying to predict a heart attack.
It sounds awful, but here's the massive aha moment. While you can't predict the specific path, you can predict the shape that the path sits on.
And this is what we call the attractor.
The attractor. Think of it like a messy room. Okay, say you have a teenager. You cannot predict exactly where a specific dirty sock is going to land on the floor. That's the trajectory. That's chaos. But you can confidently predict that all the socks will end up on the floor and none of them will be glued to the ceiling.
So the floor is the attractor.
In a basic sense, yes. The system is confined to a specific geometric shape. In chaos theory, we call this a strange attractor. It's this beautiful fractal shape. The system loops and twists around it forever, never repeating the exact same path twice, but it never ever leaves the shape.
So if we apply this to the human body, my actual heartbeat is the path.
Your heartbeat is the trajectory. The underlying physiology of your heart is the attractor. And the question we have to ask is, what does that shape look like? Is it a simple circle? Is it a complex fractal donut?
And to measure that shape, in our last deep dive, we talked about the SRB measure.
Yes, named after Sinai, Ruel, and Bowen. This is the crucial link between geometry and statistics. You can't predict the specific weather event. You don't know if it's going to rain next Tuesday at 2 p.m. But the attractor determines the climate, the long-term averages. The SRB measure is what tells us those statistics. And biology, as we are going to see today, cares deeply about the SRB measure. It is how your body maintains stability even when all your individual cells are acting totally crazy.
And then finally, we talked about the Feigenbaum cascade. This was the universal route to chaos.
This one is so critical for today. Robert May showed this first with ecological models. As you stress a system out, like, say, increasing the growth rate of a fish population in a pond, the system doesn't just instantly go crazy, it doubles. Period 1 becomes period 2, then period 4, then period 8.
It's like a stepping stone process.
It's a cascade.
Yeah.
And the rate of this acceleration is governed by the Feigenbaum constant, 4.669. This number is universal. It applies to fluids, it applies to electronic circuits, it applies to fish, and today we are going to see that it applies to flesh and blood.
Okay, the foundation is laid. Let's jump into Act 1 of our outline, the heart.
The engine room.
Now, I think most people, and honestly myself included before reading these sources, have this assumption that a healthy heart is a metronome. It just goes tick, tick, perfectly regular. If I check my pulse right now and it's skipping around, I'm probably calling 911.
And, you know, that intuition is completely logical, but it is effectively wrong.
It's wrong.
It is dangerously wrong. If your heart is beating like a perfect metronome with absolute, unyielding regularity, you are likely in very deep trouble. You might actually be approaching heart failure.
Wait, you're saying a regular heartbeat is a sign of sickness?
I'm saying that perfect regularity is a sign of a system that has lost all its flexibility. The heart isn't a single ticking clock. It's actually a massive network of coupled oscillators. You've got the sinoatrial node, the AV node, the Purkinje fibers, the muscle tissue itself, and your autonomic nervous system is constantly modulating all of that. But let's back up to how the heart actually breaks. Let's talk about how we get from a normal beat to that bag of worms fibrillation we saw in the ER.
Okay, walk me through it.
This is where we see the Feigenbaum cascade in action inside the human body. And I want to be clear about our confidence level here. This is Tier 1 science. Solid, replicated experimental evidence.
Tier 1, so we
Right.
can take this to the bank.
Rock solid. Researchers like James Weiss and Alan Garfinkel at UCLA demonstrated this beautifully in 1999. Imagine you stress a heart. Maybe you cut off the oxygen flow a little bit, ischemia, or you introduce a drug toxicity like too much adrenaline.
You're just turning up the pressure on the system.
Exactly. At first, the heart stays in period 1. Beat, beat, beat. Every single wave looks identical on the ECG,
That's normal sinus rhythm.
right? But if you turn that stress knob up, the heart doesn't just jump straight to chaos. It splits. It goes into period 2.
And what does period 2 actually look like on a monitor?
It's a pattern of alternating strength. Strong, weak, strong, weak. If you looked at an ECG, you'd literally see a tall T wave, then a short T wave, tall, short. Cardiologists have a name for this. They call it alternans.
Alternans. Strong, weak, strong, weak.
Got it. Now turn the stress knob up just a little bit more. It splits again, period 4. Now the heart cycles through 4 distinct morphological patterns before repeating. Strong, weak, medium, weird. Strong, weak, medium, weird.
Like a wobbly wheel in a grocery cart, just getting wobblier.
And then it hits period 8. Then period 16. The splits happen faster and faster. And here's the thing that should honestly make the hair on your arms stand up.
I am ready. Lay it on me.
If you measure the precise rate at which these splits happen, meaning how much extra stress you need to add to get from period 2 to period 4 versus period 4 to period 8, it follows a very specific mathematical ratio.
Don't tell me.
It is the number 4.669, the Feigenbaum constant.
Why is that so spooky to me?
Because think about a dripping faucet. You know when a faucet is just dripping steadily, and then you turn the handle just a tiny fraction of a millimeter and the drops start to go drip, drip, drip?
Yeah, the rhythm changes. It groups up.
That faucet follows the exact same ratio, 4.669, if you heat up liquid helium in a convection tank, 4.669. If you model the population growth of a certain species of fish, 4.669.
That is just bizarre. You're telling me my heart failure follows the exact same mathematical schedule as my leaky bathroom tap.
I'm saying that the route to chaos is universal. It does not matter if the medium is fluid or fish or flesh and blood. The math underlying the transition is identical. The heart is literally doing advanced nonlinear calculus as it dies.
Wow. So the end of that road after period 8, period 16, period 33 today, the final destination is chaos.
The final destination is ventricular fibrillation, total aperiodicity. And here's the really crucial twist. The heart is 3D. It's not just a single point in time. It is a thick chunk of meat.
Right. It has actual volume and depth.
So when that rhythm shatters, it doesn't just shatter in time, it shatters in space. This is what Gillian-Quay and his colleagues modeled in 2000. It's a concept called spatiotemporal chaos.
Spatiotemporal. So the alternans pattern, that strong weak thing we just talked about, that's happening across the physical space of the heart too?
Yes. Initially, the whole heart does it together. That's concordant alternans. The whole tissue does a strong beat, then the whole tissue does a weak beat. But as the cascade progresses, you get discordant alternans.
Meaning they get out of sync.
Exactly. One patch of the heart muscle is doing a strong beat, while the patch right next door to it is doing a weak beat.
So you have neighbors physically fighting each other.
Yes, and it creates these incredibly steep electrical gradients between the regions. And those gradients cause the electrical waves traveling through the heart to break. Imagine a physical wave at the beach hitting a jetty and crashing. In the heart, when an electrical wave breaks, it curls in on itself. It forms a spiral. And then that spiral can break into more spirals. You get this absolute storm of breaking spiral waves. That is the physical reality of ventricular fibrillation.
Which perfectly moves us into Act 1B from our sources, the topology of death, because you just brought up spirals.
Yes. And this is honestly one of the most beautiful and terrifying discoveries in this entire field. It comes from the work of Arthur Winfrey.
Tell us about Winfrey, because he wasn't a standard cardiologist, was he?
It's not at all. Arthur Winfrey was this incredibly brilliant theoretical biologist. While everyone else in the field was looking at individual cells and tiny ion channels, he was looking at donuts and coffee cups. He was looking at topology. And in 1994, he published a paper in Science that completely changed how we view this. He showed that fibrillation is not just random electrical noise. It is highly organized spatiotemporal chaos with a very specific topological structure.
Topology again. We touched on this in a previous deep dive, but how does the math of shapes apply to a heart attack?
Think about those 2D spiral waves I just mentioned. If you look at a spiral, like a little plastic pinwheel, there is a specific point dead in the center, the pivot point.
The eye of the storm.
In cardiac dynamics, we call this a phase singularity.
Unpack that for me. What exactly is a phase singularity?
Let's imagine the cardiac cycle as a circle of color. Red is the upstroke when the cell fires. Blue is the resting phase. Yellow is the recovery phase. As that electrical wave rotates around the tissue, all those colors spin around the center. But at the very center point itself, it's like the North Pole on a globe. What time zone is the North Pole in?
All of them. Or, I guess, none of them.
Exactly. The phase is undefined. At that singular point, every single phase of the heartbeat meets. The cell, trapped at that singularity, has no idea what to do. Doesn't know whether to fire or rest. It is a literal topological defect in the electrical field. Now remember what we said. The heart is 3D.
Right.
So if you extend that flat 2D spiral down into a thick slab of ventricular muscle, it becomes a scroll wave. It looks exactly like a tornado.
A tornado inside your chest.
And that tiny point singularity in 2D becomes a singular filament in 3D. A string of completely undefined electrical phase threading its way through your heart muscle.
That is just a terrifying visual. A tornado made of broken electricity threading through the heart.
Fibrillation is simply the interaction of these filaments. They dance around, they tangle up, they break, and they reconnect. Your survival entirely depends on the topology. Do those filaments bump into each other and annihilate, leading to spontaneous recovery? Or do they multiply and fill the space? If they multiply, you die.
So bringing this all the way back to our cold open, the sledgehammer, the defibrillator, why does it actually work, topologically speaking?
It works through a process called topological erasure.
Topological erasure.
Think about it. When you hit the heart with 200 joules, you force every single cell in that tissue to the exact same phase, fully depolarized, at the exact same instant.
You essentially scream at everyone in the room to shut up at the same time.
Precisely. You wipe the entire slate clean. Since a singularity requires a gradient of different phases to exist, it desperately needs the red right next to the blue, right next to the yellow to maintain that spin. If everybody is suddenly forced into the red phase, the singularity just vanishes. Poof. You haven't fixed the underlying pathology that caused it, but you have successfully erased the topology.
You deleted the tornadoes.
We deleted the tornadoes by just flattening the entire landscape.
Which brings us back to the whisper, the experiment at UCLA with the rabbit tissue. Because they definitely didn't flatten the landscape.
No, they didn't. This is Act 1C, chaos control. Garfinkel, Weiss, and their team utilized a method called OGY. It's named after the three physicists who developed it, Ott, Grubogey, and York. The fundamental premise of OGY is fascinating. It says that a chaotic attractor is messy, yes, but embedded deep inside that mess are infinitely many unstable periodic orbits.
Unstable periodic orbits. Give me a good analogy for that.
Think of them like invisible ghost tracks hidden in the chaos. The system actually wants to settle into a regular rhythm, a nice, neat loop, but the loop is fundamentally unstable. So the system approaches the track, rides it for a split second, and then immediately flies off into the chaotic mess again. The chaos is constantly visiting these ghost tracks naturally.
So the order is actually hiding inside the chaos.
Precisely. Chaos contains its own control mechanisms. The OGY method says, just wait, watch the trajectory carefully, wait for it to naturally wander close to one of these stable ghost tracks, and just as it's about to fly off, give it a nudge. A tiny, tiny perturbation, just enough to push it back squarely onto the track.
So you're not forcing the system, you're steering it.
You are steering it. Think about driving a car on an icy road. If you slam on the brakes, the sledgehammer, you spin out completely. But if you tap the steering wheel gently at the exact right moment, the whisper, you regain control. Why? Because the ice is slippery. The system is highly sensitive to small inputs.
That implies that chaos, in a weird way, is actually a more flexible and responsive state than order.
Yes, and that perfectly transitions us to the second major finding in the heart, one that completely flips our standard medical intuition upside down. This bridges us from Tier 1 into Tier 1-2 territory. This is the work of Robert Kliger and his colleagues from 1987. They were looking at heart rate variability, or HRV.
HRV. I hear about this all the time now, mostly with fitness trackers and smartwatches. High HRV is supposed to be good, right?
High HRV is excellent. Kliger studied a group of patients who had survived heart attacks, and he found that the patients whose hearts beat with perfect metronomic regularity, very low variability, were significantly more likely to die in the follow-up period. The patients whose hearts had irregular, bouncy, highly variable rhythms, they survived.
So the metronome heart, the one we think of as perfect, is actually the dying heart.
Correct. And here's the dynamical systems interpretation of why that is. A healthy heart operates on a high-dimensional attractor. It's incredibly fractal, it's rich, it has so many degrees of freedom, it can adapt instantly to its environment. You stand up, it speeds up, you sneeze, it pauses, you get scared, it races. That extreme flexibility comes directly from its chaotic, high-dimensional nature.
And the sick heart.
Pathology is a collapse of dimension. As the heart gets sick, as the tissue scars, as the coupling degrades, as the autonomic nervous system fails, it loses those degrees of freedom. It becomes rigid. It becomes regular. It degrades into a simple, low-dimensional machine. You don't die because you become chaotic. You die because you lose the high-dimensional chaos that actually allows you to adapt and be alive.
That is just, it's profound. We constantly equate order with health, but biology equates order with rigidity and death.
Complexity is health. Simplicity is death.
Okay, we've looked deeply at the heart. We've seen the Feigenbaum cascade, the topological tornadoes, the whisper of control, the HRV. Now I want to move up the body. Let's talk about the brain.
Okay, and right here I need to place a flag. A very big, very red flag for the listener. We're officially moving from Act 1 into Act 2.
What's the flag for? Why the shift?
Because in the heart, the physics is tier 1. It is rock solid. We can visually see and replicate those scroll waves in a petri dish. But the brain is infinitely messier. We're entering tier 2 territory now. The measurements we have are real, but the interpretations of those measurements are much hotter, much more heavily contested. We need to maintain our intellectual honesty here.
Got it. Tier 2. Proceed with caution. So we're talking about seizures, epilepsy.
Right. If you look at an EEG, an electroencephalogram, a brainwave recording during an epileptic seizure, it looks exactly like an electrical storm. You see these massive high-amplitude spikes, wild crashing waves. To the naked eye, it looks like total unmitigated chaos.
It looks like the absolute definition of disorder. If I showed that chart to anyone on the street, they'd say, that brain is going completely haywire.
Exactly. But in 1986, researchers Babloyans and Dasex decided to apply the mathematical tools of chaos theory to these specific EEG recordings. They used the data to reconstruct the geometric attractor of the seizure, and they found something that completely shocked the neuroscience world. The seizure is not high-dimensional chaos. It is actually low-dimensional.
Wait, the massive electrical storm is low dimension?
Yes. Think about normal, healthy wakefulness. Right now, you are listening to me, you're aware of the temperature of the room, you're maybe tapping your foot, you're thinking about what questions to ask next. Your brain has billions of neurons firing in these incredibly complex, semi-independent, desynchronized patterns. That is high dimension. Lots and lots of degrees of freedom.
It's like a really busy, complex conversation, like a massive cocktail party where everyone is having 100 different nuanced chats.
Beautiful analogy. Now, a seizure is fundamentally a state of hypersynchronization. Suddenly, all those individual neurons stop having their own conversations and they lock together. They all start chanting the exact same word in perfect unison. Fire, rest, fire, rest. The overall complexity collapses. The degrees of freedom just vanish. The brain gets trapped in a highly simple, highly repetitive, high-amplitude loop.
So just like the heart, the disease state is actually a collapse of complexity. It's a massive simplification of the system.
Precisely. The seizure is a dimension collapse, and this realization directly led to what we call the Lennard's prediction. In 1998, Lennard's and Elger showed that you can sometimes actually see this drop in mathematical dimension before the physical seizure even starts.
Seriously?
Yes, the attractor starts to actively shrink, the complexity slowly drains away from the EEG signal, and then the actual seizure hits.
I mean, that sounds like an incredible diagnostic tool. Can we predict seizures this way? Like give patients a warning?
That is the holy grail of epilepsy research. The answer right now is sometimes. It's just not reliable enough yet for a clinical device that you'd confidently implant in someone's head today. And this brings us right to the essential critique we have to cover. We have to mention James Thiele.
Right. Who is Thiele?
James Thiele is the physicist who basically walked into the chaos party in 1986 and said, hold on a minute, everyone. Let's look at the math. He published a landmark paper on something called spurious dimension. See, calculated the dimension of an attractor requires a massive amount of incredibly clean data.
And brain data is, well, messy.
It's notoriously messy. EEG data is incredibly noisy, the recordings are often short, and the brain changes its state constantly. It is highly non-stationary. Thiele rigorously showed that if you aren't extremely careful, colored noise, basically just random fuzz that happens to have some internal correlation, can actually fool your fancy algorithm into falsely reporting a low dimension.
Oh, wow. So you might think you found this profound chaotic attractor, but you really just found a random pattern in the static.
Exactly. He mathematically proved that just because your computer spits out a number and says the dimension is 2.5 doesn't actually mean the system is operated on a 2.5-dimensional strange attractor. This critique was a cold shower, but it forced the entire field to grow up. They had to develop rigorous statistical controls, like surrogate data testing.
What's surrogate data testing?
It's basically where you take the real data, mathematically shuffle it to completely destroy any underlying geometric structure, but keep the basic statistics the same. Then you run your algorithm again. If it still says low dimension, you know your algorithm is being fooled by noise. You use the surrogate to prove the structure in the real data is actually real.
Okay, so where do we stand today then? Is the whole seizure finding real, or was it just noise?
The current consensus is that the changes in dimension are real. The transitions are robust. Normal wakefulness is undeniably a higher dimension than sleep. Sleep is a higher dimension than a seizure. But the absolute numbers, the exact dimensionality, we take those with a massive grain of salt. We don't hang our hat on the specific digits anymore, but the trend of dimension collapse during pathology is robust.
Okay, so the brain, just like the heart, desperately seems to need this high dimensional complexity to function healthily, but why? Like, fundamentally, why would the brain want to be chaotic?
And this brings us to one of the most evocative, beautiful concepts in all of complexity science, the edge of chaos. This is Act 2B.
I love that term, the edge of chaos. It sounds like a title for a sci-fi novel.
It does. It originally comes from computation theory, specifically the work of Christopher Langton in 1990. He was studying cellular automata, these simple grid-based computer simulations. He had this parameter, lambda, which was basically a knob he could turn from pure order on one side to pure chaos on the other.
And what did he find?
He found that if the rules are too strict, a low lambda, the system freezes. It becomes static. No information can move across the grid. That is pure order.
So it's like ice.
Exactly, ice. Now, if you turn the knob the other way and the rules are too loose, the system just goes totally crazy. Random, turbulent noise. Information is destroyed the second it's created. That is chaos.
And that's like vapor, gas.
Right, so you obviously can't build a functioning computer out of ice because nothing moves. And you can't build it out of vapor because nothing persists. But Langton found that right at the boundary, the extremely narrow phase transition between order and chaos, you get what he called class 4 behavior. You suddenly get these complex, beautiful structures gliders that can persist in their shape but also move across the grid. You can store information and you can transmit it. Langton forcefully argued that universal computation, the actual ability to process complex information, is mathematically maximized at the edge of chaos.
And so the hypothesis here is that the human brain actively tunes itself to sit on this exact edge.
Yes. That is known as the criticality hypothesis. In 2003, researchers John Beggs and Dietmar Plenz looked at actual physical slices of rat cortex. They measured something called neuronal avalanches, cascades of neurons firing in sequences. And they found that these avalanches follow a perfect power law distribution.
Power laws. We've definitely seen these before in our E9 deep dive. Small events are super common, massive events are incredibly rare, but there's no typical scale to any of it, right?
And power laws are the absolute, undeniable fingerprint of a system that is sitting at a critical point, just like the famous sandpile model we discussed. The functional argument here is profound. The brain maintains itself exactly at the edge of chaos, because that is where it is mathematically most sensitive. A tiny input, a single whisper in a quiet room, a single photon hitting your retina can propagate through the entire neural network and be processed without instantly dying out in order or exploding into a massive seizure of nonsense and chaos.
It perfectly balances stability for memory and sensitivity for perception.
It uses the SRB measure to turn wildly unpredictable individual neuron firings into completely reliable, stable perception. The brain is forever walking a tightrope over the abyss.
Man, okay, we've done the heart, the organ scale, we've done the brain, the tissue scale, let's go all the way down. Act 2C, the cell, the genes.
The smallest scale. We go to Stuart Kaufman, 1993. Kaufman is a giant in this space.
Yeah.
He treated gene regulatory networks basically like computer logic gates. Genes turn each other on and off in these massive tangled webs. He built models of these called Boolean networks.
And did he find the exact same regimes Langton found?
He did. He found the ordered regime where genes freeze up and the cell basically dies.
Yeah.
He found the chaotic regime where genes flip on and off randomly, which looks a lot like cancer and genomic instability. And he found the critical regime, the edge of chaos. But here is the absolute kicker, the numerical coincidence that still keeps theoretical biologists up at night.
Let's hear it.
At the critical boundary, exactly at that edge of chaos, Kaufman found that the number of stable attractors in the entire gene network mathematically scales as the square root of the number of total genes, n.
The square root of n. Okay, let's just do the math live right now. How many genes do we actually have?
Humans have roughly 25,000 protein coding genes.
Okay, 25,000. So the square root of 25,000 is exactly 158 point something, so roughly 160.
Right. Roughly 160 mathematical attractors. Now, biologically speaking, how many distinct cell types are there in the human body? You've got liver cells, neurons, skin cells, red blood cells.
I've heard estimates usually around 200 or 300.
Between 200 and 300.
Wow.
The numbers match incredibly well. Kaufman's model powerfully suggests that cell types are not magic. They are simply the physical manifestation of the attractors of the gene regulatory network operating right at the edge of chaos. Sui Huang expanded on this beautifully in 2005. A cell fate isn't a pre-programmed destiny. It's a basin of attraction.
That is just uncanny. You have 25,000 genes, and the abstract math of chaos theory accurately predicts exactly how many different ways those 25,000 genes can stably combine to make a human body.
It implies that cell type isn't a random biological list we cataloged. It's a mathematical necessity dictated by network dynamics. The edge of chaos provides just enough stability for a liver cell to stay a liver cell. It stays trapped in its attractor. But just enough flexibility and diversity to build an entire complex organism from a single fertilized egg.
So if we pull back and look at the total synthesis of all this material today, we have essentially built a ladder.
A ladder of chaos spanning every scale of biology.
Let's recap the rungs. Rung 1, gene networks. Attractors at the critical boundary literally define who we are. They create our cell types.
Rung 2. The heart. High-dimensional chaos allows for instant flexibility and adaptation. The collapse of that dimension leads straight down the Feigenbaum cascade into the topological tornadoes of fibrillation.
Rung 3, the brain. Normal consciousness lives dynamically on a high-dimensional attractor. Seizures are the terrifying collapse into low-dimensional hypersynchrony, while universal computation hums along perfectly at the edge of chaos.
And Rung 4. Ecosystems. From our previous deep dive, the SRB measure allows animal populations to persist over decades despite wildly chaotic environmental fluctuations.
It's the exact same pattern, everywhere, at every single level of existence.
And that is the unifying theme. Life does not fight chaos. Life actively uses the geometry of chaos. It builds its reliability entirely on top of the SRB measure. The individual trajectory of a cell or a heartbeat is wild and unpredictable, but the statistics, the attractor, is solid. And the dimension, that single mathematically calculable number, is the ultimate diagnostic. High dimension is health, adaptation, and life. Low dimension is pathology, rigidity, and death.
You know, this whole discussion raises a really massive question for me. We've talked about this word dimension in so many different ways across our deep dives. We have the fractal dimension of coastlines and clouds. We have the Fisher information rank from our episodes on statistical models. We have the attractor dimension here in dynamic biology. Are these all just the exact same thing?
Now, that is the open question, the deep dive speculation, if you will.
Go for it. Speculate.
It is entirely possible that these are all just measuring the exact same underlying fundamental quantity, that the geometry of fractals, the statistics of Fisher information, and the dynamics of strange attractors are literally just three different languages for describing the exact same hidden property of complex systems. I have to stress, it is highly suggestive, but is not mathematically proven yet. But if it is true, if we can link them, it means there is a grand unified theory of biological chaos waiting to be written.
A grand unified theory of life. That is wild.
It is the absolute frontier of the science right now.
Amazing. Okay, before we close this out, let's do our standard confidence review. You always keep us honest. How solid is all the stuff we talked about today?
I will score it for you. Let's start with Tier 1, the solid rock. The cardiac period doubling cascade, the presence of the Feigenbaum constant in failing heart tissue, Winfrey's 3D scroll waves, Garfinkel's OUI chaos controlled in the lab, and Kliger's finding that low heart rate variability predicts mortality. These are hard facts. They are highly replicated.
Excellent. And tier 2, the probable but messy stuff.
The brain research. The observation that EEG dimension drops during a seizure is real, but as Thaler showed us, the specific numerical dimensions are highly sensitive to noise and heavily debated. Seizure prediction via dimension drop is incredibly promising, but still hit or miss clinically. The whole edge of chaos hypothesis for brain computation and Kaufman's square root of n scaling for gene networks. These are beautiful, mathematically compelling theories supported by some experimental evidence, but they are not universally accepted, proven laws yet. They are tier 2.
And the open question.
The convergence of all these different dimensional measures. That is purely for the future. We'll have to see where the math takes us.
Okay, so what are the 3 big takeaways for the listener to carry around in their head after listening to this?
Takeaway 1. Life uses chaos. Biology intentionally operates near the chaotic regime to gain statistical reliability. It surfs the attractor rather than fighting it.
Love that.
Takeaway 2. Chaos is steerable. Because a chaotic system is, by definition, highly sensitive to initial conditions, you don't actually need a sledgehammer to fix it. If you know the geometry, you can use a whisper.
And takeaway 3.
Dimension is diagnostic. The loss of fractal dimension, the loss of complexity, is the universal mathematical signature of disease. When the complexity collapses, the biological system fails.
So good. Do you have a final reflection for us?
I just keep thinking about the defibrillator from our cold open. It saves lives, absolutely. We need it. But conceptually, it is a relic. It is a blunt instrument from an era before we knew the heart actually had a shape.
Before we knew about the singular filaments and the tornadoes.
Exactly. And I truly believe that someday we will have sensors fast enough and computers smart enough to read the geometry of a fibrillating heart in real time. We will mathematically map the attractor while the patient is on the gurney. And instead of hitting them with 200 joules of arc welding energy, we will just deliver a tiny, perfectly timed millivolt nudge. We will finally trade the sledgehammer for the key.
That's incredible. And it actually leaves me with a final provocative thought for everyone listening. If a loss of dimension is the signature of disease in a heart or a brain, what if that's what aging is? What if getting older isn't just wear and tear, but a slow, system-wide collapse of our fractal dimension? A slow descent into rigidity. Something to think about.
That is a chilling but fascinating thought.
Well, on that note, keep your attractor strange and your dimensions high. Thanks for taking this deep dive with us.
See you next time.